Hey everybody, so enzymes. Exactly what are enzymes? Well, enzymes are a type of protein. So let's check this out. Most enzymes are proteins. They're tertiary or quaternary structures, so they're those complex type of proteins that we looked at in the organic macromolecule unit. Now, they act as what is called a catalyst to accelerate a reaction. Okay, a catalyst is the scientific term for a compound that actually speeds up whether compounds form or whether compounds are broken up. They're not permanently changed in the process, so they can be reused and recycled and used over and over and over again. Now, they are very, very specific for what they catalyze. Okay? Because they are reusable, they can be utilized in body's reactions over and over, multiple times, and very, very quickly. Now, we know something is an enzyme because it always ends in the word ASE, okay? So sucrase is an example. Lactase is an example. Maltase is an example. When we look at an enzyme, we can see that that quaternary structure has this opening, or it has a special site called the active site. Now, because it acts as a catalyst, it works on what is called a substrate. And the substrate is whatever that compound can chemically fit. So the active site is going to be very, very specific and attached, just like a lock and a key, or I'm sorry, a key and a lock, with the substrate. Now, enzymes work by weakening bonds, which actually lower the activation energy for a chemical reaction to occur. So when I'm looking at this diagram, this is what's difficult for most people to understand. Many reactions, you have your two reactants, and in order for them to combine and form a compound, for a bond to form, there's going to be a difference. Sorry, my mouse keeps disappearing. There's going to be an input of energy in order for that reaction to occur before it can release energy and form a bond and form the product of that reaction. So the difference here is the amount of energy that's required. That is what we call the activation energy. Circle this in your packet. Circle that right there. And label it as activation energy. It's the amount, again, that is required for that reaction to occur, and that is what the enzymes help with, is it decreases that amount of energy. Now, here's another example. Let's look at a graph of a chemical equation without an enzyme. So do you see how the line goes up, and it goes up fairly significantly? Okay, it goes above what we call the free energy of activation. And when we add the enzyme to the equation, put it in the mix, it actually lowers the amount of energy. So remember, the more we go up this y-axis, the more energy is required. You can see with the pinkish purple line that the activation energy doesn't go up as high. So there are different enzymes and reactions that occur. And so when we look at this, we can see enzyme here that has no substrate added to it. Okay? So nothing is happening. But as soon as we add the enzyme and the substrate, okay, which in this case happens to be liver homogenate, okay, the liver homogenate and the hydrogen peroxide react pretty rapidly, pretty fast, and creates all of those bubbles. Now that's the product that ends up being formed. Okay, so let's take a look at the actual substance, or what we call the enzyme substrate complex. Okay, so we've got the big enzyme. You've got this picture at the bottom of your page. Okay, the enzyme itself acts on a substrate. at what we call the active site, and that's where it joins. So the active site, now this is at the top of the next page, is a restricted region of an enzyme, and the molecule binds to the substrate at that point. I'm sorry, we're not at the top of the next page, we're still on the previous page. So it causes a change in the shape of an enzyme. We call this, and I know that this is cut off at the top of your screen, we call this induced fit. UCED, fit. This is what we consider to be induced by the substrate. So when we look at that enzyme, sorry, I went backwards. So that induced fit, causes a change in the configuration of an enzyme's active site. This is where hydrogen ions and ionic bonds are involved. This can be induced by the substrate itself. So when we look at the enzyme, we have that substrate that fits in there, and that's what we call the induced fit. Okay, so what exactly affects the enzyme activity within an organism? There are three different factors. First, it could be environmental conditions. Second, we're going to talk about cofactors and coenzymes. And third, we're going to talk about enzyme inhibitors. So let's look at the environmental conditions first. Extreme temperatures are actually the most dangerous to enzymes, and that's because proteins are affected by temperature. If they get too hot, their structure actually unfolds and unwinds, and the protein doesn't even fit anymore. The bonds technically have come apart, and that compound doesn't even look like the same compound anymore. So high temps do what we say is called denaturation, and they denature, or they unfold that enzyme. pH is another environmental factor. Most enzymes prefer a fairly neutral pH, close to a pH of 7, so anywhere from 6 to 8 is pretty safe for them, but outside of that range, not so good. They begin to break down. Then there's ionic concentration. So if there is a really high salt concentration, there's not very much water present, that can also affect the activity of an enzyme. Now, let's take a look at cofactors and coenzymes. These are considered inorganic substances. Okay, inorganic meaning they don't have carbon in them, they're not produced by living things, so things like zinc and iron, and even sometimes vitamins, respectively, sometimes are in need for proper enzymatic activity. So iron has to be present in the quaternary structure, that really complex structure for hemoglobin on your red blood cells, in order for it to even pick up the oxygen. If there's not enough iron, then you're not going to be able to carry as much oxygen, you're going to be tired. This is what we call, when you find out if somebody's anemic, somebody has low iron in their bloodstream, that means they're not getting enough oxygen, their hemoglobin isn't functioning properly. So there are two different examples of what we call enzyme inhibitors. There is what's called competitive inhibitors. And if you think about what an inhibitor actually is, an inhibitor is going to prevent something from happening, right? So a competitive inhibitor is a chemical that would resemble the normal substrate and actually compete with it for the active site. So when we're looking at the enzyme and we're looking at that structure that exists on the active site, a competitive inhibitor is something that's going to be shaped fairly similar or look very close like the substrate, and that enzyme is going to actually attach to the competitive inhibitor instead of the substrate. So no good there, because that means our chemical reaction isn't going to occur. Another kind is what we call a non-competitive inhibitor. These inhibitors don't actually go on to the active site, but they bind to another part of the enzyme, causing the enzyme to change its shape, which in turn alters the active site. So this time when we look at the enzyme, notice how the active site already looks a little bit different. So the non-competitive inhibitor is actually going and attaching on the opposite side of the enzyme. Over here is where the active site used to be. Now it's been altered. It looks completely different and in no way, shape, or form would be able to bind to its substrate. All right, that's it for enzymes, and that is the final piece of our organic biochemistry notes.